Hello, welcome to week 1 laboratory. I'll go over the laboratory objectives, and then I go over a little bit about the lecture PowerPoints just as you're going through them, some things to look for when it's going to help you with your weekly assignment. So today I'm doing week 1 blood. Okay. In the laboratory, we're really going to focus on the cells, or what we call formed elements, and then also a little bit on blood typing. The lecture PowerPoint is going to be Chapter 19, and you're definitely going to want to study your PowerPoints. That's a lot of what gets tested on the lecture exam. Okay, so how about your blood lab manual? So we're going to be doing cardiovascular system blood, so we're going to be doing that. And so we'll start off with part 1. And so you'll be able to go through these objectives, but basically this is what we need to accomplish in lab today, or this week for you. So, blood lab. First of all, blood. Blood is made up of about 45% formed elements and about 55% plasma. Now, formed elements, you call them cells, they're not all cells, because with the formed elements, we have 3 different types of formed elements. 2 of them are cells. One is a cell fragment. So the first one we'll look at is we have our erythrocytes. Here's our erythrocyte, otherwise known as a red blood cell. Red blood cell has no nucleus, actually. It's made in the red bone marrow. Right before it's released, the nucleus is extruded, and then we have this cell. Since it doesn't have a nucleus, doesn't have any DNA, cannot actually make proteins, so its lifespan is limited. We're talking about like 120 days in circulation. So you have to constantly make a lot of them. Your blood is 45% formed elements. It's like 44 point something, pretty high, 44.9 just about percent erythrocytes. So if you look at blood is red. You have the erythrocytes. That's most of the formed elements. 1% of the formed elements, or less than 1% of the formed elements, are going to be your other ones like your leukocytes and platelets. Okay. All right. Well, the leukocytes are the white blood cells, and there are 5 major types of white blood cells. Right here, this is a platelet. A platelet, sometimes called a thrombocyte. We have a bunch of platelets here. We got 1, 2, 3, 4, 5, 6, like 10 platelets here or so. These little wires here, these are strands of fibrin. Now, fibrin is a clotting factor. It's found in plasma as fibrinogen. Fibrinogen is soluble in the plasma, and then when the clot forms, the fibrinogen becomes fibrin. Fibrin is insoluble and then forms a clot. Okay. These are platelets here. Okay, so we have platelets, erythrocyte, otherwise known as red blood cell. Okay. Then we have all our leukocytes. Now, our leukocytes, there are 2 major categories or classifications of leukocytes. We have our granulocytes and we have our agranulocytes. The granulocytes have granules in them, and they end in fill, like for what type of stain they like. And so, for example, the most common leukocyte. So if you were to look under the microscope, you'd see a whole bunch of red cells. You might see some platelets, little flecks, these like purpley colored flecks in there. And then you'll see every once in a while you'll find a white cell. Now in the white cell or leukocyte, the major type of granulocyte you'd have is a neutrophil. This is a neutrophil here. Okay. It has very small granules. The one thing that you notice about it is it has really this funky multi-lobed nucleus. And so if the multi-lobed nucleus, that's really what what defines it. And also it's fairly common when you see it. So you have neutrophil. We have an eosinophil. Eosinophil loves the eosin stain, so it has these red granules. The eosinophil is fairly uncommon. For example, we have neutrophils. 75% or so of the white cells are going to be neutrophils versus like 4 or 5% would be these erythrocytes. The most rare white cell called a basophil. Okay, so here's a basophil. Basophil likes the basic stain, has these dark granules in it. Okay, very hard to find. You're talking about like 1% of 1/10 of 1%. So very, very rare. Usually, like if I were to find this under a microscope, I got to hunt for a couple minutes to try to find one of these basophils. Okay. All right. So we have our granulocytes. We got our neutrophils, eosinophils, basophils. Okay. And then we have our agranulocytes. Agranulocytes don't have granules. We have two different types. We have right here. Okay, this one here, this is a lymphocyte. See, a lymphocyte is fairly small, usually a big nucleus, very, very little cytoplasm, and so sometimes you just see a little ring of cytoplasm. Okay. Then we got the monocytes. Monocytes are huge. Now, the monocytes, they call these monocytes in blood. These can migrate out of the blood into the connective tissue, and when they're found in the connective tissue, these are called macrophages. Sometimes these cells, they'll move in and out of blood, and they get different names. So the early histologists basically named cells in connective tissue, named cells in blood, and then found out it was actually these cells were the same cell, actually. And so when we look at these, as far as like what these cells do, okay, we have the neutrophils most common. So if there's going to be some type of infection, they're usually the first to be there. Okay? Versus like monocytes, these are the big cleanup crew. That is going to come in later. And so a lot of times with an infection, you'd see the neutrophils first, and then a lot of dead neutrophils, dead bacteria or whatever it's trying to fight, and then you'd see the the heavy artillery come in here, the macrophages start cleaning up a lot of the tissue. Macrophage. This is a monocyte. Okay, here is a agranulocyte. Okay. We have our eosinophils. The eosinophils are going to be found elevated when you have parasitic worm infections, those kind of things. They also can break down some of the effects of the basophils. The basophils, okay, they are going to be involved in the allergic type of reaction. Let's say these things, they release histamine and heparin. Histamines are released when you have some kind of pathogen in your body. The basophils are going to do it. So if you have an allergic reaction, allergies acting up, you might see elevated numbers of basophils. Okay. And we have our lymphocytes. Lymphocytes are found, basically, they are involved with the immune system, and so we'll talk more about these when we get to chapter 22 in immunology. I think that's on the second test, okay. Third test. Okay. But later in the semester. But they are involved in immunity. All right, so those are all the cell types, all the functions. Make sure you know what's what, be able to identify. Okay. Let's see. Now we finished our blood. Here, we talked about the cell types. As I'm going on here, okay? So you can pause it. You can take those notes there. So we talked about functions of the cell. Here, this differential. Now, you don't need to know exact numbers of the cells. What I want you to get an idea is basically kind of relative numbers. So if you've got a differential white cell count, which means normally you do just a simple blood cell count. If your leukocytes come in elevated, then you would order a differential white cell count. Differential white cell count would actually look at what cell types are elevated. Now, normally, you're going to have the most neutrophils. Okay. And the order that it goes in is never let monkeys eat bananas. Okay. And so, never let monkeys eat bananas. Normally, it's neutrophil, lymphocyte. Okay. Never let monkeys, monocyte. Okay. Eosinophil. Okay. Eat. And then basophil is most rare. That's typical. But let's say if you're having that like allergic reaction, you might see elevated numbers of basophils. So instead of never let monkeys eat bananas, it might be never let bananas monkeys eat. Okay, something like that. Okay. All right, so make sure you kind of have an idea of what relative numbers, what's more or less. Okay. All right, so that's differential. Then also hematocrit. What is hematocrit? Hematocrit is percent of formed elements. Now I said before you have 45% formed elements, 55% plasma. That's normal situation. In that case, your hematocrit would be 45. If you were at 40% formed elements, 60% plasma, then your hematocrit would be 40. Okay, very simple. Typically, your hemoglobin is going to be correlated with your hematocrit. So that with the hemoglobin, let's say if your hematocrit is, let's say 45, then you kind of divide it by 3 to get a rough estimation of your hemoglobin. So if your hematocrit's 45, your hemoglobin would be like 15, okay, something like that. If your hematocrit is 30, which is pretty low, but not necessarily critically low, then your hemoglobin would be 10. And so it just depends on your physiology, what's going on with you. Typically 30, there's some kind of problem. That's not typically normal. This is basically what I know about blood. I'm a reproductive physiologist. My experience is more in biology, that kind of more laboratory kind of thing. Any kind of clinical experience, I have to ask my expert who I live with. My wife is a board-certified anesthesiologist who works at Metro Health Medical Center as a major trauma center, level 1 trauma center in Cleveland, Ohio. They have Life Flight, and a lot of times people will show up on Life Flight, show up to the operating room, and she'll get people with hematocrits in the 20s and have to put in a lot of blood products very, very quickly. So what she tells me is that they can be walking, talking with a hematocrit of 30, but usually they're not doing so well. A lot of times if their hematocrit gets down to like 35 or something like that, they wouldn't even necessarily transfuse if they're stable, conscious, that kind of thing. Okay. All right. Those people will be very, very anemic, but they might be okay. All right. So that's hematocrit. Just give you a little bit of background on hematocrit. Okay. Now, blood typing. Okay. Because if you get a patient who's going to need blood, you can't just put any blood in there. Okay, into them. There will be a reaction. The reason why is because on the surface of all of your red blood cells, okay, I can show you the note here. On the surface of all of the red blood cells, okay, you have antigens. Okay, antigens are basically protein or glycoproteins, and you have genes that determine what antigens are on the surface of your red blood cells. Okay. Now, your types of antigens, you might assume are A, B, and O. They're actually A, B, and RH. O is a type, and that's actually the empty set, okay, which means you don't have an A or a B. So, for example, I'm type O positive. I don't have A or B on the surface of my red cells, but I do have the RH antigen, and the RH antigen is marked by a positive or negative. So it's like I am O, so I don't have A or B, but I am positive for RH. My wife, she's an A positive. My brother is also A positive. That would be, they have the A antigen, they have the RH antigen. Okay. Now, whatever antigens you have on the surface of your blood cells, that determines your type. However, you have the opposite, what we call antibodies, are going to be floating around in your plasma. So over time, we pick up antibodies to every antigen that's not actually ours. So then, where we have it says antigen, opposite antibodies in the plasma. So for example, if me is O positive, I just have the RH antigen. So that means in my plasma, I have anti-A and anti-B. Okay? Which means if you give me my wife's blood, okay, then I would form a transfusion reaction. I'd have a transfusion reaction to the A antigen because I would perceive that as foreign. Okay? They say that people who are type O negative would be the universal donor because they would have no antigens on the surface of their cells. But the thing is that when you're donating whole blood, you're also donating the antibodies, so that causes problems because someone with type O negative would have the A, the B, and the Rh antibodies, and so that would be a problem. When, say, someone who is O positive, O negative, a lot of times they like them to donate whole blood, and they can use the different products and also use the red cells, pack red cells, not use the plasma, because then that would not have the antigens. Someone who has a rare blood type like B negative, or even someone like if they are AB positive, someone who's AB positive, sometimes they say they're the universal recipient. It's not necessarily true. You could have transfusion reactions because of the antibodies. However, that person would probably want to be a plasma donor because an AB positive would not have any antigens in their plasma. Okay. Now, what I'm going to demonstrate for you, and I kind of just have this all set up so that our video, I try to keep them like under 20 minutes, is basically we have a lab where we do some typing. Okay? So now for lecture, okay, and even lab practical, make sure you memorize this. Okay? Alright, so match that up. And I will ask you questions on basically like blood would have these antigens, what blood type, or someone would have this blood type, what antigens are present. I'm also going to do it here, which I'm going to just see if this works. Okay. I shoot this down like that. Okay. You see those? Hi. Okay. And we'll mix these up. Okay. What I did was basically each well plate is a person's blood. So for example, this is Mr. Brown's blood here. So we have Mr. Brown, Mr. Green, Mr. Jones, Mr. Smith. Okay, so these are our 4 patients, and so the 4 patients' blood are the well plates. And then in each well in the plate, you have an A, an RH, and a B. Okay, so I know it's upside down. Okay, say, let me turn them around, I guess, for you. Okay, and so you can see them here. Okay, is that good? Okay, and what I did was then in each plate, you see if the A's, okay, then I have an anti-A antibody, and I put A in all those, okay, and then I have an anti-RH, I put RH in all the RH wells, and I put B in all the B wells, okay. Then kind of just stir them up with a just basically with the toothpick, and kind of just stir them up and see if there's any kind of reaction. Reaction would be it's kind of where it kind of gets chunky and cloudy. You see this one's starting to react up like that. Okay. And so what's happening is, okay, look at this one here. What's happening is what we call agglutination. So the antibodies can agglutinate the antigens, okay, and cells, and actually cause the cells to get all clumpy, and even cause the cells to burst. Okay. And kind of mix them up so you can kind of see what's going on. Okay. And so basically, you see as they're mixing up. Okay. Here's the lab. Okay. Hopefully under here you can kind of see a difference. You see how it's a little bit lighter under the video? This is like what I'm seeing. Okay. And I can point what's going on. Okay. Right here. Okay. Well, actually, I should probably start with this one for you, so you're as you're looking at it. Okay. This one right here. We have A, RH, and B. Okay. These are the possible antigens. We had a reaction in A, we had a reaction in RH. That means we have the A antigen and the RH antigen. So they have A, they have RH, and then you determine their type. This person's type would be A positive. Right here, we only have a reaction in B, we don't have a reaction in A and RH. So this person has the B antigen, and their type would be B negative. This one, you see a reaction in all three, which means they have all three antigens, A, B, and RH, and then their type would be AB positive. Okay? Over here, no reactions. They have no antigens, and so when we added the antibodies, there was no reaction. And so when they have no antigens, then their type is type O negative. Okay? And so that's how you do the blood typing, and so that's a major part of this lab. Okay. All right. Now, the last thing I want to talk about is something like you'll go through your PowerPoints in lecture, and you'll see this, where it's plasma versus serum. Okay? And so I want you to basically, you can pause it or take the notes on this, okay, on what's the difference between plasma and serum. Okay? So, what's the difference between plasma and serum? Plasma has clotting factors, serum does not. Okay. And so what's happening? Well, the major clotting factor that we're talking about is going to be fibrinogen. Fibrinogen is soluble, and so it's soluble in plasma. When you form a clot, it becomes insoluble, and then that's fibrin. So what happens when you clot is you have extrinsic factors and intrinsic factors. Okay? Factors released by platelets, factors released by the damaged endothelium, of basically damaged blood vessels, the cells of blood vessels, okay, would release these damaged blood vessels, and the platelets would release these factors, and they would create a cascade of reactions to basically cause fibrinogen to be converted to fibrin, and that forms the clot. So how do we get plasma versus how do we get serum? Well, first of all, why would you want plasma or why would you want serum? Well, plasma you'd use as a blood product that would have those clotting factors, so you want to put that in somewhere. However, serum is really good to use in a laboratory. Like, for example, I was studying sea urchin sperm. I use seawater, but sometimes in some laboratories, if they were studying some kind of like mammalian sperm or something, they might want to use serum for their cell culture. And a lot of times cells can be cultured, even other cell types, liver cells or something like that, they could be cultured or grown in a dish. The dish would contain serum. You would want the cells to grow in the serum. You would not want to use plasma, because if you use plasma, you see this agglutination, what you saw in the in the blood typing, that would start to happen with your cells. So then you have this big gooey mess, and so you don't want that. So, for example, with my, let's say if I had bull sperm, which we had in our lab, some people were doing studies, experiments on that, they'd want to use serum, not plasma. Serum wouldn't get all chunky. So how do you get the serum away from the plasma? Well, they do have serum separator tubes, but I'm going to tell you, just basically kind of explain to it the old-fashioned way, which kind of gives you a little bit more of the science of what's going on. In order to get plasma, what you want to do is you want to take whole blood, and you put it in a centrifuge. A centrifuge is basically a machine that spins. Centrifugal force will separate out the formed elements from the plasma, because the formed elements are heavier, and so they would sink to the, what we call the bottom, or the pellet in the blood. So you can see these, look in your book, look in your PowerPoint notes, you can see hematocrit, how it can separate plasma from formed elements. So you can take whole blood, prevent it from clotting, spin it in a centrifuge, and then you will get plasma on the top, which they call the supernatant, and you have formed elements in the pellet. To get serum, what you need to do is you let the blood clot first. When you let the blood clot, the fibrinogen comes out of the plasma. It's going to mix in with the formed elements and form a clot. So then when you centrifuge it after it's clotted, then you would have, in the supernatant, you would have serum, and then you would have, in the pellet, you'd have the clot, which would contain your formed elements and your fibrin, okay, which is pulled down out of the plasma. So that's the difference between plasma and serum. You'll see there's an open space in your PowerPoint notes that you can put those notes in as you're studying. Okay. that actually concludes the laboratory lecture video. I kind of combined it for this week, and so study this video, study your PowerPoints. If there's something in your PowerPoints, make sure you look at the, look in your book for explanations. If you need help, always send me an email. And so this is basically how it's going to work. Look for some your weekly assignment, which will basically be questions, short answers, discussion, dealing with this video, and you should be able to answer those questions. So, thank you very much. We'll see you next week.